Welcome back, everyone. I hope you enjoy your lunch, your short lunch, and like me, you're probably still eating it. So it's my pleasure to introduce our next agenda item, and I will turn it over to Dr. Daryl Zeldin, who will introduce our next speaker.
So thank you, Gary, and good afternoon. So it's a pleasure today to introduce the DIR speaker, Dr. Patricia Jensen. So Dr. Jensen is head of our developmental neurobiology group in the neurobiology laboratory in DIR. She received her PhD at the Neuroscience Institute at the University of Tennessee Health Science Center in 2002 and completed postdoctoral training at St. Jude Children's Research Hospital and also at Harvard Medical School before we recruited her to NAEHS in October of 2009. So Dr. Jensen has become an internationally recognized scientist in the fields of developmental neurobiology and also catecholamine biology. Her research is focused on the development and function of the noradrenergic neurons in health and disease. These investigations make use of mouse models combined with cellular, molecular, behavioral, and in vivo systems neuroscience approaches to provide insight into the mechanistic relationships between environmental exposures during development and increased susceptibility to emotional and cognitive deficits later in life. Studies from her group have opened new avenues of research into the development and function of the noradrenergic nervous system, leading to paradigm shifts in norepinephrine biology. In addition, her group has designed and generated a series of viral and recombinase-mediated genetic strategies in mice that are being employed by neuroscientists here at NIEHS and the broader scientific community and all around the world, really. And in recognition of these accomplishments, she was promoted to senior investigator with tenure in 2019. So today her talk, as you see, is titled The Neuroadrenergic System and Environmental Health Science, and I'll turn things over to Patricia. Thank you. So Patricia, I think you're muted. There we go.
Okay, great, thank you. They turned over control to me and I can no longer see the unmute button. So thank you, Daryl, for the introduction. Shake your head if you can hear me now, hopefully. Great. It's already started moving forward. All right, we're gonna get this show going. All right, so thank you, everyone. Good afternoon. It's really a pleasure to be able to present some of my work for you today. What I hope to do over the next 30 minutes is to first tell you a little bit of an overview of our research program here in DIR. And then for the remainder of the time, to tell you some of the results of our latest study in the lab. So to begin with, I want to just tell you a little bit of, what is the neuroadrenergic system? All right, so the neuroadrenergic system are the neurons in the brain that actively synthesize norepinephrine. This is a really interesting population of neurons in that it's a very small subset of cells. So if you think about it, there are estimated to be about 90 billion with a B neurons in the human brain, and these neurons are made up of about 100,000 neurons. And then they're also situated really deep within the brainstem. But even though it's such a small population in the brain, their impact on our behavior is just amazing. And that's due to their long axonal projections that are highly collateralized, right? So multiple branches, so one neuron can communicate with different parts of the brain. And given this, it's known that norepinephrine in the adult is very important for a number of important physiological functions, including things like attention, mood, appetite, memory, and our response to stress. Given that function, it's not surprising that these neurons are also affected in numerous neurodegenerative disorders, as well as developmental disorders, and also following exposure to, a wide array of different environmental insults. What caught my attention when I started studying these cells is that the data that shows that this vulnerability to disease-related cell death and environmental insult is in just subsets of these neurons, really. So really showing this differential vulnerability. And the best example of that is in Alzheimer's disease and also in Parkinson's, where these neurons are some of the first to die. And it's just a subset of the neurons in the largest cell group here in the pons, known as the Locus Ceruleus. All right, and data from rodent models, there's also some intriguing evidence showing that that same differential vulnerability exists. And so one study I'm just going to point out here is it was a mouse study with prenatal exposure to 1-bromopropane, and it affected the axons going to the prefrontal cortex, but not to the hippocampus. So this really highlighted the differential vulnerability of these neurons. And so given this vulnerability, we feel that the key to understanding this function of these neurons in disease won't come from studying the system as a whole, but instead will come from really uncovering the genetic factors that define unique functional subtypes of neuroadrenergic neurons. So to address these questions, the long-term goal of the lab is to really understand the mechanistic relationship between perturbation of very distinct subtypes of these neurons during early development. The primary focus on the lab has been during prenatal development, and how that may affect an increased susceptibility to deficits in emotion and cognitive behavior. So to do this, we use the mouse as a model system. Just like in the human brain, these neurons are located in the brainstem here. So in the pons, the LC, you'll see throughout my talk, is the locus ceruleus. There's also the subceruleus. And then these medullary populations, the A2 and the A1 area, that are much less studied than the locus ceruleus. And, I always show this image because it's just a really easy representation of the system. But if I zoom in on what the system really looks like, so I'm going to take a cross section through here. And what you'll see is these are actually bilateral populations of neurons, the same in the human brain. And so this is the cerebellum here and the fourth ventricle. And the locus ceruleus sits right here on either side of the ventricle, the fourth ventricle here, right? And it's nicely compact, and that makes it much easier to study than these other populations that are really scattered, really going from the whole dorsal to ventral surface. And the same if you look back at these A2 and A1 neurons in the medulla, they're really scattered. They're super hard to study. And so given our interest in really knowing what these neurons are doing developmentally, really called for a new way to look at the system. And as Dr. Korshutz said yesterday, generation of new tools to be able to ask these really important basic biological questions are everything. And so a lot of our work is developing mouse lines that really allow me for the first time to ask questions about the development of these populations. And so the way we do that and the way we start it, since diversity within any population really starts early in development. we knew that the area where these neurons arise in development is the hindbrain, and that hindbrain early in development is segmented into different molecular domains called rhomomeres. And since we knew genes that were differentially expressed there, we use those to fate map the neurons and see where do they end up in the adult, right? So we could express fluorescent markers very early and very selectively in these different neuroadrenergic neuron domains. And so what I'm showing you here is an example where I can label these neurons that we call HOXB1 neurons, because early in development, they express the transcription factor HOXB1. It's a very scattered population that labels part of the pontine nuclei, and then scattered populations in both the A1 and the A2 nucleus in the medulla. Right, and so once we do that, we can then use that same fluorescent markers to start asking things about what's the function of these neurons, right? So the first key to that is, where do they project? So what other regions of the brain do they communicate with to direct behavior? And so what I'm showing here is the results from really these 3 studies, is showing that the locus ceruleus, which we know provides this amazing input to really almost every area of the brain and spinal cord. But this other population, HOXB1, also provides just this tremendous projection, including to some areas of the cortex. So prior to this original study, it was believed that the locus ceruleus provides the only input to the cortex. Because these 2 populations project to areas that we know are involved in response to stress, and we knew very much that the locus ceruleus is important in the stress response, and my lab and others have shown that if you directly activate those neurons, you get an increased anxiety-like response, increased depressive-like behavior. So we then generated a mouse line where we could selectively activate these 2 populations at different times. So for instance, when I went in and activated these HOXB1 neurons and looked at the response behaviorally to an acute stressor, like a forced swim, or put in a well-lit arena, these neurons did the complete opposite of the locus ceruleus. So instead of initiating that anxiety-related behavior, we got a decrease in anxiety-related behavior. We also saw things like a decrease in heart rate. So it was a really exciting study. And so once we have these things nailed down, where we have information about where these neurons sit, what is their function, then we can go in to the next step and say, okay, when I disrupt these developmentally, what's going on long-term? And there's several aspects to these studies. So one is just asking, what is norepinephrine doing early in development? So norepinephrine is in the brain really early. In the human brain, it's detected in the developing neocortex by 8 weeks of gestation. In the mouse by embryonic day 14, which is mid-gestation. So before the brain is even completely formed, you see norepinephrine. So I can use these same tools to start asking what norepinephrine is doing there. And 2 of the ongoing studies in the lab is, one, just doing that prenatal selective disruption of the locus ceruleus during development. It's the first time that anybody's been able to do that, and looking at the long-term consequences. And a study that fell out of that is just our foray into doing incorporating environmental exposures. So we started a study looking at prenatal chlorpyrifos exposure. And the reason we did that is there's a ton of epidemiological data showing that prenatal exposure to organophosphate pesticides do affect things like learning and memory and social deficits. Well, that is also our phenotype when we disrupt norepinephrine from the locus ceruleus prenatally. So studies are ongoing in the lab, really trying to piece together, what's going on during those early time events, and what are the long-term consequences. Like I said, those studies are really in their infancy, but hopefully I'll be invited back to tell you the results of those studies in the future. For today, I want to tell you about the latest study in the lab, which is the role of the locus ceruleus in the modulation of feeding. This is work that was conducted by Natalie Sciolino while she was a postdoc in the lab. This work laid the basis for her K99 R00, and she's just in transition now, starting her lab in the Department of Physiology and Neurobiology at the University of Connecticut. And so we know that norepinephrine plays a role in feeding. A number of the drugs that are given for overeating affect the levels of norepinephrine. They increase the levels of norepinephrine. But the majority of work looking at the neuroadrenergic system has focused on these centers here in the medulla, the A2 region. And those are the neurons that likely are communicating directly with the gut. But Natalie, being just a fantastic postdoc, paid close attention to some of the older data looking at the role of the locus ceruleus. So there's these set of seminal papers looking at the function of the locus ceruleus. And this one from 1981 from Gary Aston Jones and Floyd Bloom was really the first to show that the locus ceruleus is responsive to sensory stimuli, right? So this is a classic paper. It's one that everybody cites. And so what you're looking here is a readout of the excitability of the cells here upon a sensory stimuli. And the one I'm showing you here is a tactile stimuli, like a touch. And you'll see as soon as that happens, these neurons become active. And it's through that activity that they release norepinephrine downstream areas. Right, and so this was then followed by this decrease in activity. And that was the same for, visual stimuli, auditory stimuli. But Natalie paid attention to one figure in the paper. There was one animal that was done where they looked at a rat that was licking sucrose. And it was sort of a throwaway line in the paper that what they observed was this decrease in activity here. So the complete opposite of the other sensory stimuli, a decrease, then followed by this sharp increase here that then was maintained. So just based on this piece of data, Natalie asked the question, does locus ceruleus activity modulate feeding? And so the first experiment she wanted to do was to look at the activity patterns of these locus ceruleus neuroadrenergic neurons during feeding behavior, right? And so the way we can do that now is a technique known as in vivo fiber photometry. I'm really lucky. I just started incorporating this technique in the lab. And I'm lucky because it was really pioneered by my colleague, Guohong Sui, who's a tenure track investigator in neurobiology. And this is a way of imaging calcium fluctuations as a readout of cellular activity while the animal is freely behaving. So the way we do that is we, in this case, we generated a mouse line where we're expressing FLIP recombinase under the control of dopamine beta hydroxylase, a gene that's important for the conversion of dopamine to norepinephrine. So we get selective expression in neuroadrenergic neurons. We then directly deliver a calcium sensor via a viral vector right into the locus ceruleus, stereotactic bilateral injections. And you'll see the orientation in the virus is in the reverse orientation. And that's because it requires flip recombinase to recombine the allele. So it's rotated in its place, and then some of the flip recognition sites are removed, so it stays in place. And so then we can hook the animals up to a fiber optic cable, spectrometer, and a laser, and look at the calcium fluctuations as the animal is eating. And so we can do this in a time-locked fashion. So what I'm going to show you here is the system we use. It's called a FED, a feeding experimental device, that was developed by Lex Kravitz, who's now at the university, at WashU now, but he developed this when he was at the NIDDK. And so you can see here the mouse that's hooked up to the fiber optic cable. And I just want to show you that these mice, they're fine. We set them up in this before we actually do the recordings and see that they can move freely. They readily approach this pellet dispensing area. So where a pellet is dispersed, every minute the animal immediately approaches it, picks up the pellet, starts consuming it. And so we can time lock the calcium imaging to this behavior. And so the data we get, we can represent in multiple ways. So what I'm showing you here is a raster plot for showing you one animal and what it looks like across the session. So for these animals, they're fasted overnight. So they're ready to eat when you're doing the experiment. And so what you're looking here is that every row is a pellet that they're consuming across time. Right, and the color is indicative of the calcium fluorescent signal. So the more red it is, the higher the activity, the bluer, the lower the activity. And then you can look at this information across the different pellets. And so what became obvious when we were looking at this, if you look at the first 10 pellets versus the last 10 pellets, what you'll see is during the first 10 pellets where the animals are approaching, you see this increase in firing of these neurons. So an increased calcium, increased activity. But as you move further in this session, as the animals become more sated, you then see this decrease, right? While the animals are consuming, you also see this decrease in activity. So another way of showing this is to show you the average across animals. And so for me, I think this is easier to show. So in this case, the fasted mice, so early in the session, this is when they're hooked up. The animals are just freely moving. The pellet drops, they approach it. You see this spike in activity. As soon as they retrieve and start consuming, you see this drop that then slowly returns back up to normal. Later in the session, as the animals become sated, you also see this drop in activity during feeding, but it's definitely attenuated. And this, what you see in the early stages to this approach, that's almost completely gone. And what I'm showing you here is the cumulative data for this, showing a difference between both the fasted and sated. And so this is a lot of animals. It's a lot of data to put together. And so Natalie followed up with a linear regression analysis to look at this. And what she sees when you look at both the approach and the consumption, so across all the animals, you see this gradual influence of satiety on these two different behaviors of the locus ceruleus neurons, both during approach and then also in consumption. Okay, so next, Natalie wanted to know, does the locus ceruleus neuroadrenergic neurons and the activity of those neurons play a direct causal role in the suppression of feeding? And so the way to test that is during this little window here, which is just a few seconds in time, if we activate the neurons there, right, when they're eating, can we interfere with feeding? Will the mice eat less when we do that? So we turn to another technique called optogenetics. In this case, we're using an opsin called Crimson R. It's a cat ion channel that we can express similar to the calcium indicator, selectively expressed in the locus ceruleus. And it's sensitive to light. So when we shine light on the neurons, we can activate the cells. And so that's exactly what Natalie did following the surgeries and the implant of the optics, fasted the mice overnight, put them back into the fed system. And then during the retrieval, or at the start of the retrieval, she stimulated the neurons. And so indeed what she found was that indeed they ate less, which was really exciting to see, that she can interfere with the actual feeding behavior during this really small, really millisecond scale. Also, there was a trend in that the animals really stopped retrieving the pellets as well. And she's looking closer at that now to see, was it different throughout, you know, from the beginning of the trials, the more fasted versus the more sated. So with this data, she next wanted to ask, okay, what's the long-term consequences of doing this? You know, using optogenetics, it's, you know, this very short term stimulation. What if you stimulated over this longer period of time? And so we did that by using another mouse line in the lab where we selectively expressed this mutated human muscarinic receptor. So this approach is called chemogenetics. So these modified receptors are no longer activated by the native ligand, acetylcholinesterase, but instead are activated by clozapine and oxide. Right, so we can selectively express these. In this case was a transgenic animal we made where we could selectively express it in the locus ceruleus, but not these other neurons, and then do the long-term studies. So the first study Natalie did was to food deprive the animals overnight, inject with vehicle or the CNO, and then after 20 minutes, put the animals into this arena to just do a food preference test over a 30-minute period and just look at the amount of food that the animal consumes during that time. And so what you see here, there was no effect of CNO on the controls, the littermate controls, but the activity of the locus ceruleus neurons did affect feeding. She then next moved on to using, doing a several day trial using a LabMaster in-cage system or home cage system where you can really look at, you know, food intake, you can look at liquid intake, and look at energy expenditure, a readout of metabolism over the time course. Right, and so what Natalie found is that, you know, sure enough, so basically the way she did this was, you know, first day was no CNO, then there was a day given 30 mg per mil in the water, then a day off of just receiving water, and then increased to at 100 micrograms per mil the CNO in the drinking water. And you can see in the where we activated the locus ceruleus, there's this significant decrease in food intake. But what she found that was interesting, that it's due to fewer meals consumed. Right, so this was significant that over the course, the animals where we were activating the locus ceruleus consumed less meals. There was no difference in the meal size, and there was no difference in metabolism. So next, Natalie wanted to look as where this effect may be mediated. So we know that the hypothalamus is the primary feeding centers in the brain. And there was showing that you can inject norepinephrine into the lateral hypothalamus, and it affects feeding. It also affects the activity of the neurons in the lateral hypothalamus. So we looked at FOS as a readout of cell activity in the hypothalamus, and sure enough found that in the lateral hypothalamus, when we activate the locus ceruleus, there's decreased activity there, but not in these other known feeding centers in the dorsal medial hypothalamus or the ventral hypothalamus. She then once again turned to optogenetics, where she stimulated this pathway. So in this case now we inject the virus into the locus ceruleus, but the light source is over the hypothalamus. And you can see here when we just stimulate the terminals in the lateral hypothalamus, you get this reduction of food intake. But we saw no effect if we inhibit that pathway. Right, so what Natalie thinks that it's more of a brake on the system versus an accelerator. We next looked at, knowing that the role of the locus ceruleus in eliciting both aversion and anxiety. We looked at that pathway in several tests of anxiety-related behaviors and aversion. And just to move it along, since I'm running out of time, is the answer is yes, that this particular pathway, we are also affecting effects of anxiety and aversion. So these animals are more anxious and more aversive to stimuli. Right, so what Natalie thinks is that the locus ceruleus modulates feeding by integrating both external cues like anxiogenic cues and internal states like satiety. So in summary of what I've shown you today is that the locus ceruleus, we've shown for the first time that activity is suppressed during natural feeding behavior. And then interestingly, that the LC activity response to food is attenuated by satiety. And that activation of this pathway to the lateral hypothalamus also suppresses feeding and promotes a negative affective behavior. What is unclear now, and is what Natalie's following up in her own lab, is what are the cell types in the locus ceruleus, so we know they're neuroadrenergic, but is it a subset? And then what are the responding cells in the lateral hypothalamus? And then another important implication of this study is that it really expands our knowledge of what the locus ceruleus does. But again, this concept that the LC plays this role in modulation of feeding by integrating both external cues in the environment and internal drives like satiety. And the follow up on this for Natalie, and a big focus of her R00 portion of her grant, is to look at the effects now of high fat food and diet induced obesity on this circuit and the outcomes, with the hope really of, are there ways to be able to manipulate specifically, say, this circuit and affect feeding and obesity without, the current modes, using these drugs that affect the entire neuroadrenergic system, which can have really serious side effects given the role of norepinephrine, both in the brain and in the peripheral nervous system. So with that, for some reason, it's not letting me advance anymore. John, it stopped on me. I'm just trying to get to my acknowledgement slide. Oh, there we go. Oh, so it skipped completely now. Anyway, I'd like to just quickly acknowledge all the folks in my group, our amazing collaborators, and just we have phenomenal resources, cores, and support services in DIR that really allowed me to do this work. So with that, I'm happy to take any questions you may have.
Dr. Jensen, thank you so much. That was a great presentation. We'll now open it up for questions. So as before, if you have a question, use the reactions function on the bottom of your screen to raise your hand. Karen.
Hi, Patricia, that was really beautiful work. I have a question about the diet-induced obesity studies that you plan. I know that for diet-induced obesity, there are various prescribed diets that are used, and they're actually made up generally with more fat rather than total calories or amount. But then sugars may. So I was just wondering, in the cells that you're studying, do sugars? Is it total calories? Is it fat, or would sugars stimulate those differentially? And do you plan to look into that?
Yeah, so that's a great question. It's one that I ask Natalie all the time. So I should say Natalie is taking this study with her. So I'm not going to be doing, I'm focusing on pesticides right now and more anxiety related behaviors with these different populations. But yeah, so I would suspect that it may be different depending on what the nutrient is. Sugar versus high fat versus just, the content of the diet. So she's definitely interested in looking at that. But her first studies for the grant were to look at high fat.
And does she have does she have an independent position now?
Yes, so she's starting her lab. I still call her my postdoc, but because she's still here because of the shutdown, I feel like she was on the job market during the shutdown. So doing interviews virtually was just crazy, but she accepted a position in the Department of Physiology and Neurobiology at the University of Connecticut.
Oh, so yeah, that's right. Great, that's great.
Yeah, she officially started there in January, but we have a manuscript that it's in revision at Nature Metabolism. And so she's here and we're working on the revisions, which is really why I picked it to talk today, because I was deep in it.
Yeah. No, that would be great. It would be great. We're interested in diet-induced obesity on genetic instability in mouse models, but it would be interesting to see. We don't really we do look at genetic instability in the brain, and it's very different than most other organs, but we look at whole brain, not specific areas. Okay. But it would be really interesting to make those comparisons. Yeah. Yeah, thank you.
Thank you. Other questions? Daryl.
I can't help to ask this. So it's not a scientific question. I'm mostly just wanting to have you tell council about how the kind of the long-term support that an intramural program offers, the emphasis on high risk, high reward research really made kind of the stuff that you do possible. So could you say a few words about how this is just perfect for intramural as opposed to the extramural world?
Yeah, definitely. So the types of tools that I make are really only being done by big programs like the Allen Brain Institute. So there's like no way I could, just take a faculty position somewhere and say, oh, I have this great idea to study this important cell population. I don't know if it's going to work, but I have to make all these do all these cool genetic tricks, and it's going to really pay off. They let me do that here. There was a little pushback along the way, Daryl. You have to admit some uncertainty, but it's really paying off now. And people are using my tools now to get at other complicated systems, not just in the brain, but in other systems. So a lot of folks in doing cardiac research, development of the lungs are using some of the tools that I generated in my lab.
Karen?
Yeah, I have a comment to that. To speak to that, I think that this sort of speaks to the R21 mechanisms. And for the extramural world, for those of us in academia who don't have that opportunity is sometimes to do expensive exploratory research that may really pay off. I think maybe thinking about, more R21 unsolicited or specific RFAs, and maybe a little larger, but some of these studies, I'm sure that these could not be done with the $275,000 budget on an R21. So I don't know, just something to think about us in the extramural world.
Yeah, no, definitely. So one thing that I've been really good about is I make my mouse lines and viral vectors or any of our plasmids available. So everything gets deposited into the Jackson labs, and it's been really nice. They want my line, so there's no cost to us. And we deposit everything at Addgene, for just that reason. So one thing I want to mention, because it was discussed yesterday and I feel like I should talk about it, and I just ran out of time. So the sort of my foray into doing more environmental health, neurotox work, really came out of my interest in that, similar phenotype seen. But at the same time, Dr. Wojcik, had put out for within NIEHS funding for proposals that bring together people from NTP, DIRT, and DIR. And so I wrote a proposal because I thought it was perfect for this. I was already seeking out folks from NTP who do the toxicology. And so I wrote a proposal about bridging the gap between environmental health sciences and neuroscience, and really training the next generation of scientists, who have expertise in both the fields. And so I'm really excited about this because the project has really taken off. And so in October, I have 2 new postdocs coming working on this project. So it's really exciting. And so, in thinking about that and even funding in the extramural, the importance about bringing together people from different disciplines and working as teams, to answer some of these really big questions.
Yeah, that's outstanding. And there you go, Karen. She's providing some of her tools to reduce
Right.
some of the cost of doing research.
That's excellent. Thumbs up.
So any other questions, comments? Well, I have a comment or question, Dr. Jensen. You mentioned the cross-divisional work, and I think that's really important. Can you talk a little bit about the project that you proposed and how you plan to bring the different groups together?
Yes, definitely. So, unfortunately, this started at the beginning. We started the project right before the shutdown, right? So we put together, so I had already contacted Mamta Beal, who's a developmental neurotoxicologist in the NTP, saying, we're interested in looking at this pesticide and we need help, designing the experiment, what's the correct route of exposure, and so on. And then the proposal came out. And so we put together a team of Mamta, myself, and John Hollander, who's a program officer. And so the idea is to train a postdoc and mentor them throughout the whole process. So they have good influence on both the neuroscience side, the toxicology side, but also doing, relevant research to the funding of NIEHS. And so the hope for those postdocs that are interested in, writing K99s, really working with them through that process. And yeah, like I said, we're just taking off really now. And I have 2 new postdocs coming into the project.
It's great. Thank you, Dr. Jensen. Any other questions, comments? Terrence.
Tisha, thank you so much. Very fascinating work. I'm wondering if you folks looking forward have any intention to look at single cell transcriptomics or epigenetic modulation with these tools, using things like these spatial techniques now that are available for that. It's really fascinating.
Yeah, so great question, and we're definitely doing that. So I was really naive in the beginning, thinking, okay, so I can label these cells fluorescently, and I had written proposals from day one, like to do single cell profiling. These cells are so hard to get at. So I can dissect them out, and I can get like about 600 healthy cells from the dissection. But what I can't do is separate them out from the 30,000 glia and other cells in the culture dish. And, putting them through a FACS sorter and things like that, the cells are way too fragile. So we're taking two approaches now, doing the nuclear single cell profiling, and then also the hopes of the spatial transcriptomics. To get at these populations and just really look how they change throughout development. So for instance, the Locus Ceruleus, we know we can already subdivide them with really early developmental markers, right? And label two different populations based on two different transcription factors. So to follow that throughout development and see how those lineages maybe diverge or not diverge, and then to look at functionally. So that's definitely going on. It's much harder. It sounds simple, but I'm hoping with these new strategies, we're finally going to be able to do that. And then the epigenetics is something that I hadn't really thought about till recently. So our first experiment getting into the disruption of just the Locus Ceruleus, we simply made a flox allele of dopamine beta hydroxylase, right? And then crossed to those different early transcription factors. And so what was really a pain, but now I see as fortuitous, is that the LOXP site was put in an enhancer region. And so, and the enhancer is really interesting. So it's in the new genome build, it shows up as a predicted region. What's fascinating is that brain norepinephrine is affected, but not the periphery. Right. So that's really interesting. If you think a lot, people taking plasma and looking as a readout of what's going on in the brain, that may not tell you everything. So we're trying to figure out now how to really look at what the enhancer is and what's binding to it and giving that differential regulation.
Fabulous.
Yeah, thank you. Thank you very much, Patricia. Outstanding presentation. And thank you, Daryl. We're at the home stretch now, so I'll turn the podium back over to Rick.
Great. Okay. Thanks, Gary, and thanks, Tricia. That was absolutely terrific. Always good to punctuate our meetings with a little bit of science from our intramural program. So let's move on now to a topic that I think all of you know is a very high priority for me, the issues around diversity, equity, and inclusion. And as I mentioned yesterday, we have formed a working group of council, and Karen Vasquez has very graciously agreed to chair that group. And Karen, so I understand you're going to give us a little update on where you are on things. We didn't hear from you last time because I think you were in the middle of working your way through out of a flood.
Out of that and a snowstorm in Austin, Texas. Yeah. So
Right.
that's right.
Anyways, great to have you here today. And then you've been working pretty hard. You've got some members from members that you can introduce that I see that they are on our Zoom session. And so actually I'll turn the virtual podium over to you. So take it away.
Well, thank you. I think I have 3 slides. I don't have control of the slides. Maybe John, Nathan can, oh, there they are. So I have 3 slides, so it's very brief. What I'd like to spend most of the time on today for the remainder of my allowed time is for your input, feedback, comments, suggestions from the entire group, from everyone. As you know from the February meeting, for those of you who were there, Pat presented this new working group, and Pat and Rick had invited me to serve as chair, and I'm very honored. It's a privilege to be able to serve on this working group that's really so important in so many ways. As Gary said, it's very broad reaching. It's a big project. So we would like all the input we can possibly have to make whatever difference we can in 2 years. So let me start with, I think this is the second slide. Can we go back to the first slide? John.
Yep.
Thank you.
Yep.
Thank you. Thank you. So Pat presented this slide in February, but for those of you who weren't here, and just as a reminder, our charge is actually the charge of the working group, and we have a charter in place that Pat drafted, and the group has had edited it very little. It was in great shape. So we sort of know what our direction is, though it's very broad. But how we are going to work is we get together as a group, as our working group, and we provide advice to council. And then council provides advice to the leadership, including Rick, to make whatever decisions they may based on suggestions and advice that we may provide. So that's really our charge. And examples are listed here, ideas for increasing diversity in the workforce. And this is focused on environmental health sciences research, but both intramural and extramural. For example, we're going to be looking at funding both intramural, extramural, identification of factors that may cause inequity in funding across racial and ethnic groups, etc. So if we could have the next slide, I'd just like to show you. So Pat and I've been working hard behind the scenes, and then we have this really amazing working group right now with 10 members currently, and they've all agreed to serve 2-year terms. This is fantastic. We have a really diverse and outstanding group of people here. First, we have current or former members of council. I'm a current member of council, so that's me. And then we also have in that group is Jose Cudero, Edith Parker, who's here, Jose's here as well, and Andy Shee, who was currently on council. And we also felt we should have not only council members, extramural researchers, intramural from NAEHS, but also industry and government. So we have academia, industry, and government represented. And we are very fortunate to have Dr. George Daston with us, who has agreed to join us. He was the previous, not currently, I think he just stepped down. He was the previous president of the Society of Toxicology, for example. So he's very familiar with the toxicological research going on, both in industry, government, academia. He works at Procter & Gamble. He's been there many years, but he's also affiliated with academia. So he's just a really great addition to have a different perspective. And then we have representatives from within NAEHS. And we're really pleased to have Mary Diaz-Santana, who is a postdoc in biostatistics and computational biology. And she's already provided a lot of feedback we've had 2 meetings with this group, and she has provided a lot of feedback. But not only that, because of her background and training, we think she might be useful in sort of helping us to organize all of these large data sets that we will have to search through in order to identify where we have weaknesses and where we can improve. And then Fred Tyson, of course, and he's here today. He is the program officer at DIRT. Nicole Popovich, who's also here today. I can't say enough about Nicole. She is the management analyst at DIRT, but she has agreed to be sort of our administrative support person on this working group, which is so very helpful. She puts together notes from each meeting. She gives us our sort of our working orders and what we have to do, and keeps us on track and organizes our meeting. And I can't thank you enough for that, Nicole. And then of course, Gary, we know Gary, who's the acting director at DIRT, and Pat, who's the deputy director. Gary and Pat are also part of this group, a bit sort of ex officio. That's what they say. Pat says he wants to eventually rotate out, but we don't want that. We want Pat to stay. So Pat's really the driving force of this group. We have 5 nominations currently under review. So we're hoping for a total membership of between 12 and 15, so that it's large enough to get some work done, but it's small enough to be manageable. And with that, I would like to thank everybody who's agreed to work on this committee, and those who, the new members that will come on board, we can introduce you to at the next council meeting. But if we could just have the last slide, this is really where I'd like to spend all of the time. I would like your feedback, any comments in the chat here, emailing us. This is sort of what we have come up with in our 2 meetings that we've had with the group of 10 on this working group. We think because there's so much work to do in this area, and we're just a few people, over a 2-year time frame, just providing advice. We thought that the most efficient use of our time and effort may be to divide into subgroups that address, each subgroup will address issues of racism, diversity, equity, inclusion. They will address all of those issues, but within a theme, within a subgroup. And what we've come up with so far is, well, let me go back. So what we're thinking is there will be 12 to 15 of us. So within each subgroup, we could have 2 to 3 or 4 people focused on that. And then they can meet as subgroups, smaller groups in their various subgroup topic. And then we all meet, we're meeting, our plans to meet monthly unless we need to do more than that. And then we all come back and present to each other and see how because a lot of these subgroups will have a lot of overlapping issues, of course, so we can work together and sort of divide and conquer. That's our goal. If anybody has a better idea than that, I would love to hear it. I think this may work. We haven't started to divide into subgroups yet because we haven't assembled our entire working group yet. But the 4 areas that we came up with as a group for the subgroups would be workforce. And of course, that's critical. That's for hiring, retention, diversity of the workforce, sort of how the workforce feels about these issues, things like that. Number 2, very important, obviously, is funding. And this includes both extra and intramural funding, new initiatives put out by NAEHS, funding not only in who are we funding, but what research are we funding? And then number 3 is trainees. Very important, of course, mentoring. How are they being mentored? Who is being mentored and by whom? What areas of study are they focusing on? And how does that fit into NAEHS mission and vision? What are the makeups of study sections? What about T32 grants, R01 diversity supplements? We heard a bit about that today. Well, not just for R01, for other grants for diversity supplements, et cetera. So that's a big and important area, trainees that we felt should be separated out from just funding or workforce. Finally, and very importantly, community engagement. So who is the research benefiting? So we're all taxpayers, we all pay our tax dollars into NIH and we all get benefits from that. But how can we better engage with the community in a way that also represents sort of our DEAI mission and goals, as well as NIEHS mission and goals? So that's all I really have to present today, because like I said, we're still working on finalizing our working group, but I would really like just some open conversation and discussion, advice from all of you, any of you, on how we can best achieve some kind of impact in this area, because we're all putting our passion and time and effort into it and want to be as impactful as we can. So with that, I will stop and let you, any comments, questions, advice, input, I would appreciate very much. Thank you.
Karen, I think there are some questions coming in about the membership. Katrina, do you want to ask your question just to get it on the table for everyone to hear?
Sure thing. Thank you. So Karen, thank you for your work in leading this. I know that that's always a lot more work than it sounds like, and it's a really difficult issue to slice. Like how do you break it down into manageable chunks? So since you weren't at the last meeting, we did have a conversation about how there were designated representatives in academia, industry, and government, and community was pretty striking by its absence. So it sounds like you're making efforts to get that represented. I'd just love to see that added to that list in addition to represented in the group. And then in terms of the structure, I'd like to hear the thinking, and I'm trying to get my head around the community engagement bucket. But to me, that is a part of each of the first 3 groups in such a big way. And how you structure yourselves is obviously up to you, but I can see that it's sort of a 2-way conversation about how each of those issues, each of those first 3 categories interact with community engagement.
Yes, so actually that's a really good, as you said, this is a big sort of difficulty. It's a qualitative sort of section of many things that cross one another and overlap. So you are correct that it would be included in all of the above. We put it as would funding, for example. So funding impacts mentorship, mentees, funding impacts the research and all of them. We were trying to break it down in the way that we felt we best could based on the expertise on the committee. And we do have 2 of our 5, at least 2, Pat, correct me if I'm wrong, but at least 2 of our 5 nominees that are being reviewed right now are,
at least 2.
2 are very, we brought them in exactly for that, for the community engagement component. And so I think that we felt if one of them sort of served on a community, if they both agree, if one of them serve on the community engagement sort of subgroup and one on another subgroup, because these subgroups are a bit arbitrary and artificial as well. I mean, so we will all be meeting as a large group. We would just break into the subgroups because there's so much data and information that we need to move forward, because we can't move forward in just a qualitative way, really. We want to move forward in a quantitative way. We want to say, what is X number of, Y and Z, and how can we fix A, B, C based on numbers and data? And Pat and his group, they're working very hard, and Gary, to help us to obtain the information that we need to move forward in an educated way with data. And once we get that information, we may rearrange this entire, the entire process. But I appreciate your comment, and I will add the community engagement as a group that's represented, because they clearly are, and that was very important for us. We wanted to be very, of course, as this group, the one thing we want to do is be inclusive. So we're including everyone and people with expertise as best we can with the limited numbers of people that we, can really manage on the working group. But thank you for the comment. I'll keep that in mind.
Thank you. I also have a super specific one as an example under research. It's so specific, I'm going to put it in the chat. If we have time to get to it, great. If not, perhaps we can take it up offline.
Okay. Thank you.
And Karen, we have a comment from Irva. Irva, I don't know if you want me to read it or if you want to ask it or suggest it yourself. There you are.
Well, here I'm. Hi. But yeah, maybe I'll ask it because maybe it needs a little explanation. So I thought the 4 that you had up there are they really cover, I mean, yeah, as you say, there's a lot of overlap, but they are really distinct in certain ways. But I feel like there's sort of a 5th, and I don't know if it may not lend itself very much to the quantitative measures that we look for to be able to show evidence as scientists, the way we measure things. That's the thing. But I do feel that there's an overarching question of changing the culture, and every time you try to move forward in any one of these areas, there are people who sometimes can become very vocal. I mean, there are certain things that people get very emotional about. Sometimes, and then you see, when you talk about hiring, there are these patterns of people who want to hire people who look an awful lot like themselves, and no matter how much you reason with them about, but here's this other person who's just as qualified, and then they come up with reasons why they're not so as qualified, and you say, and inside you're saying, this person is really racist, but it's not really that. It's probably, and they certainly don't see it that way. So I don't know. And that there are ways in which the culture affects the way grants are reviewed and topics are reviewed. And so somehow I feel like the idea of, and really thinking hard and deeply about how do you tackle these like hidden assumptions that people walk around with all the time. And I don't know, it's a tough one, but I think it's so fundamental to this whole process that all of us are going through in our own institutions, our own departments, and our own centers and such.
Yes, that's a great point, and thank you for that. And I agree with you completely. For example, one thing that we do now in hiring is before anyone can serve on a search committee, they have to have diversity training. We have to have, for all of the applicants that come in, we have to classify if they are willing to list their, whatever, gender, race, whatever. Then we have to categorize those and see how many there were. of each group and why, if we didn't select somebody that was underrepresented, why there has to be a written explanation for that. So they're trying, but it doesn't, to your point, it doesn't really change it doesn't change the culture. And that's what we need. If we could change the culture, then we wouldn't have to worry about any of these, workshops or required training sessions, because we should all know this. So I think that changing culture is key. I think that as a working subgroup, it would be a little difficult to assess and address. So what I think I will do, and I'm writing all these things down. What I think I will do is put changing culture under each one of the subgroups that we have, because it fits into every single one. Like you said, for funding, for training, for mentorship, it fits in under everything. We just need to change the culture in general, and we will try to address that in, like you said, more of a qualitative way, but including it within each subgroup that we already have.
Yeah, thanks.
Mm-hmm.
Yeah, makes sense.
I think I saw Trevor's hand first. If you want to comment, Trevor.
Let me see if am I unmuted?
Yes.
Yes.
Thank you, Pat, for spotting my thumbs up or hands up. I just want to make sure that the groups working on this topic are working together. So Claudia Thompson and Liam O'Fallon, as you may know, are leading an initiative based in the Environmental Health Sciences Core Center program looking at this very issue, with coming up with metrics of how we can best tackle these issues. And I think it'd be very important to have a bridge with that group, because you can share things that might be of importance that you haven't thought about. One of the things that's come out of that working group that resonates with me is the idea of spheres of influence. Each of us have our spheres of influence that we can actually address as it relates to this particular topic. The sphere begins obviously with yourself, but then with your laboratory, and for the P30 centers with the P30 center program, and then what you can do at your institution. So I'm just bringing that to your attention so that you don't forget that they have an additional initiative in this area.
Absolutely. And there are, of course, many people working on this and initiatives, not only at our institutions and at NAEHS, NIH, right? And we are actually, I forgot to mention, but Skylar Walks is actually our assistant at University of Texas at Austin. She's our assistant dean for DEAI, and she is working sort of with me as sort of an advisory person to me, and she's put me in contact with a number of these groups, and that's how I first sent the mailings to get names for the working group. And we absolutely need to find a good way, a better way to bridge with other groups, as you mentioned, because there's no need to reinvent the wheel, and this is such a complex and huge problem. The more we have working on it that are thinking of metrics, for example, the better off we are. So thank you, Trevor. I may contact you later to get more information from you, if that's okay.
And I just want to mention, thanks, Trevor, for that we do have, obviously, Fred's from DRT is familiar to some extent with the P30 program, and I am, and Gary. So yeah, we will be certainly be pulling that in. And I think as we move forward, we want to be interfacing with a number of activities going on, including the Unite program that that Gary and Rick both, I think, mentioned. So as time goes on, we'd like to interface with those groups. So I think I saw George's hand. No, I think I saw Jose's hand next. So Jose, how about you?
Oh, thank you. And I just want to add to Irva's comment that I think that a lot of what may come out of this committee and recommendations, it will touch into some form of culture change. And it's really a challenge, but I think the example that Karen gave, and I on just like grant review said, or it was Katrina that also mentioned it. It's just an example, and I think that but in each of those areas, 4 categories of 4 work groups, it seems that culture change is something that will need to come. So I'm glad that we will have it as an overarching issue to be addressing. So thank you.
Yeah, George, did you want to comment?
Yeah, it's slightly different, and maybe it's something for Rick to comment on more than anything, which is the three of the four areas that we're going to concentrate on, so hiring, training, grant funding, those are really under your control, pretty much 100%. But the community, is the research serving all the communities equally, kind of thing? I think that that's probably going to go beyond NIEHS, and might be even more than cross NIH. In that we see lots of things that are probably your research combined with EPA's regulatory authority or something like that, that are going to make the difference, or your research along with research on metabolic diseases or whatever it is, or psychological stress, whatever it is that enhances or promotes adverse environmental effects. So I guess the question is, how far do you think you can push whatever it is that we come up with other agencies so that we can make a bigger splash?
Yeah. Well, actually, yeah, I think this is a good question for me. And you may all recall that one of my leadership values is collaboration. And I strongly, firmly and strongly feel that we can undertake bigger problems if we're working collaboratively together. So let's take climate change as a great example. This is going to be an issue that we will be tackling, and climate change, we will be doing this within the framework of environmental justice, but it's not going to be just NIEHS. And I'll be back in touch with all of you the next time we meet. I've actually specifically reached out to several other IC directors who are kind of independently putting together their own plans and said, look, we need to come together and develop a collective plan. And I'm hoping that eventually we'll get all of the ICs at the NIH working collectively together to figure out what it is that we can do together to address the issue of climate change and health. And then it needs to extend beyond the NIH. We need to be working together with the EPA. And actually, George, as you know, much the same as what we do with the National Toxicology Program. This is not just NIEHS, but reaching out at NCTR, NIOSH, and EPA, Department of Defense. So I can imagine that we need to be reaching out and to be working together with all of the federal agencies and nonprofit organizations that share a common mission. So I'm personally just, this goes down to my DNA, that working collaboratively together. And I think, as I mentioned to you last time, I've always run my own lab this way. It's not just doing my own thing, but it's working collaboratively with others. And I think one of the key things we need to do is provide the reward mechanisms that encourage the type of collaborative activity. So, and then overlaying all of the different elements of diversity, equity, and inclusion on the way that we do our collaborative efforts and engage in collaborative behaviors. I don't know, does that answer your question?
Yeah, it does. I mean, it's more around, is there going to be energy to push this? And it sounds like there is.
Well, absolutely. And in fact, that's, I think, part of the reason why Francis chose me as the director. I mean, this was part of my vision, the discussions that I had with him. And look, the environment is integral to everything that happens across the NIH, and it's integral to so many things that happen across the federal government. And it's not that we haven't done this in the past, it's just that I personally am going to be dedicating a lot of my energies to making sure that these collaborations can happen. And just again, overlaying on diversity, equity, and inclusion, there are just lots of things that have to be factored into the equation. It's just like, climate change, diversity, equity, and inclusion is very multifaceted. And we need to be keeping this in mind as we plan our collaborations, as we plan our science, and as we plan culture change. What's abundantly clear at the NIH, as we've been doing a lot of reflection as part of the Unite committees, that first committee is the U committee, is really understanding where do we stand. What's abundantly clear is if you look across the NIH, there has been structural racism that has been integral in our culture, and we need to fix that. So I'm really hoping that Karen, you and your colleagues on this committee It's good to see that Andy is here and George is here, and let's see, who else do we have on from the committee? Fred and others. I want that input from you, and any type of recommendations. But I'm actually also looking for very specific things. What can we do? And not just providing us with more information, but are there specific recommendations that come in? Do the following thing. I can't promise you that we'll do it, but I will promise you that I will listen to you, and I will let you know if we don't do it, why we don't do, and what are the reasons for that. So, and I also hope you will build on those 10 goals that emerged from the Unite committees. And if you need us to send them to you again, or we can give you the URL, you can get access to them. These are the things that the IC directors have decided that we're going to tackle as the phase 1 of implementing on diversity, equity, and inclusion. So lots of collaboration, focus on changing culture. And Irva, I'm so glad you brought that up. And let's just make some things happen that overall, in the end, it changes the way we treat each other. And the way that we can conduct collaborative science.
Jose, I think you have the next one.
Rick, thank you for your comment, because actually I think that you have sort of answered the question that I have. Let me just ask it anyway. And it's that this committee is one of the 10 or the number of committees that every institute and center actually at NIH would have. And the question is that it's possible that our committee, along with others, may have common issues that may need to be addressed, not at the level of each institute, but something that actually be more collectively. And and I assume, and I think that you already alluded to it, that for those issues that yes, collaboration between institutes would happen. But one of the issues that may come is sort of new ways to review proposals, for example, and may be not only NAEHS but others.
Well, Jose, and by the way, sorry, just seeing you in the upper left-hand corner, you had your hand raised. I should have noticed that. So one thing you may all recall in one of the goals was to hire a diversity officer that reports directly into the office of the director at every IC within the NIH. So I would just encourage Karen, once we have this person hired, that's someone who you may want to be interfacing, or that could be a senior leadership interface with your committee. And what I'll be asking that person to do is to make sure that the things that are happening at NIEHS are well coordinated with the things that are happening at the other ICs across the NIH. So kind of getting to that point, Jose, it's not just what happens at NIEHS, but there may be some best practices. There are things that other ICs are doing that we want to learn from. We don't have to rediscover the wheel. Let's just share information and benefit from others' experiences, most notably best practices. And I know all of your organizations are doing a lot around the issues of diversity, equity, and inclusion. So if there are things that you see that we're not doing, we'll want to make sure at future council meetings that we block out time for Karen and all of you in this committee to have a chance to report back and get these things out in the open and tell us what it is that you've been thinking about, what have you been discussing, and making recommendations to me and to the rest of leadership.
I would point out, Katrina put an interesting chat in the chat about kind of following up on Trevor's idea of spheres of influence. I think that's worth, you don't need to, I mean, you can if you'd like, but I think it kind of explains itself. I think it's a good way to look at that paradigm, kind of ever widening circles. Bob also put in a chat, I don't know if Bob, if you want to read that, or I can read it if you're.
Oh, yeah.
Why don't you read it just in case others,
okay,
it's at the table for all of us.
There he is.
Or Bob, go ahead.
Yeah.
I just wanted to, and Karen answered the question, but the idea was that we really need to do an evaluation first, because I think on a lot of these issues, there's this sort of normal human reaction to let's fix it, but we don't really know what the problem is unless we do an evaluation first. So we do need a really in-depth evaluation. I think we'll probably be surprised at some of how intransigent some of the issues are. And there are reasons behind why those issues are intransigent, and we have to really think hard about what those reasons are. And I think I'm glad this is going to be data driven, because I think there's going to be some really hard issues to address, which will also be difficult for political reasons in some cases to address, but need to be.
And I would also kind of echo that the need for data and making data-driven recommendations. One thing I would like to hear, maybe I'm speaking for the group, working group too, are ideas on how to get a handle around what kind of data we need. First off, there's a lot of data we would need, and a lot of it's going to be difficult data. So I think one of the things the working group has to work out is what do we need in the form of information, and what's a reasonable amount that we can handle, if you will.
Yeah, the advantage is that we're either scientists or working in a scientific organization. There's nothing like data to help to make decisions.
Is it something that perhaps we're too inside the box to see, and we actually need a consultant from outside to help with those issues? Because I worry that all of us we're all successful in this system. So we're invested in it. And maybe someone from the outside we need to hear their voice.
Actually, I'm going to ask our acting deputy director, Gwen. I see you're smiling, and Gwen, in her role as acting deputy director, has been doing a lot of really hard work around gathering data and figuring out how do we coordinate our diversity, equity, and inclusion efforts, anti-racism, with things that are happening across the NIH. Yeah, Gwen, do you want to take a minute and just chime in here and let the group know what you've been up to?
Sure. Thanks, Rick. Happy to add my perspective here. So a couple of things I think that you mentioned that are really important connections is that the advisory committee to the director of the NIH has two working groups, I believe, related to workforce diversity, and there's another title I can't exactly remember. And there's also the Office of Scientific Workforce that is in the Office of the Director. So those are organizations that have already deliberated around these things sort of at the larger biomedical level. They have data sources, they've written some reports. I think we will, be able to pull some of that information and those reports together as maybe an orientation package for the committee. I think what I see is that we're interested in the environmental health sciences community, and so do we believe that the data more generally collected, mirrors what we're seeing in our own communities, or do we need to move into different data collection opportunities? So some of these same issues have come up for just how the Institute is thinking about diversity, equity, and inclusion, how we're interfacing with the Unite committees, how offices like Equity, Diversity, and Inclusion, which is an office at NIH, which actually has a lot of data on the workforce at our Institute and across NIH, can help us in thinking about all these things. We've also been exploring the concept of a consultant that was a little bit internally focused at the time, but we have not yet procured those services. So listening to you talk about perhaps the need for consultant services to support this committee might be something that we can look into as well. But I appreciate all of the ideas. I did like that it was in the chat. I don't know if we pulled this out. Edith had a suggestion about focus groups with, key communities within the environmental health community. Certainly that aligns with what Unite is doing across the NIH and other communities. Like we had talked about the listening sessions for the HBCUs and minority serving institutions. So, that is a way to collect data. It's qualitative data, but important data. And if we have somebody to help us set up these focus groups in a way that's rigorous, I think that can serve the needs of the committee as well. And what I'm hearing is, there's a lot of thought work that needs to go into constructing what it is we want to ask, how we do it, what the methodologies that the committee will use. And I can tell you that I'm happy to support these activities in any way. I know DERT staff will be able to do that. And I know Rick is behind us in, you know, getting to that place where this is a committee that can really pull it together. I mean, it's almost mind blowing, right? That the number of things that we could work on and the number of things that we are facing, but we can't do everything. And so, you know, I charge the committee to think carefully about which are the most important ones to get started with, you know, what might come later in a longer term effort, and not try to do everything at once, because I don't think we'll be able to capture all of that.
Terrific. Gary, I see that you have your hand up, and I know that you've been, well, you alluded to this in your presentation, but I know that you've been doing a lot of work in DERT around the issues of diversity, equity, and inclusion.
Yes, thank you. I just wanted to comment on this issue around data and collection, and I'm glad that Edith made the comment in the chat box, and Gwen echoed that comment. When we think about data, we think about, you know, this quantitative data. It might tell you how we are doing in terms of diversity, right? And we might see things, if we like stratify the data in certain ways, we might think that, you know, we're doing well if the data are aggregated. Once we stratify them, we see that we're not doing so well in certain areas. So that can tell you one thing about what's happening in terms of diversity, but how do we get at these other metrics like equity and inclusion? And I think that you know, having these other data sources like focus groups, listening sessions will help us get there. And so I think that we need to broaden, you know, how we think about data and what type of data we need to collect here. And, you know, as I mentioned, Karen put me as a member of the working group, and, you know, I really don't want it to be too DERT heavy, but I'm happy to sit in meetings. I usually do, and I'm here to offer support and resources, you know, as you need them. I'm happy to make connections with other things that are happening across NIH. Provide resources in terms of, you know, our equity, diversity, and inclusion office, maybe provide speakers. The other thing I wanted to mention is, you know, I think we need to be on the same page in terms of what we mean by, you know, racism and, you know, having racist policies or an environment that's racist. And I don't think that people really understand what that means. I think folks tend to make it, you know, not looking at the systematic things, but, you know, saying something racist is somehow saying something about them as an individual. And I'd like to get away from that kind of thinking, right? And think about the systems that we have in place that may be racist, that may provide a structure or a barrier for people from certain racial ethnic groups to be involved in networks, for example, where they're exposed to, you know, investigators who have data sources that they could use to propel their career, or high performing scientists, you know, within academic environments who have a great deal of grants that they could participate on and in that way propel their career. So, you know, and perhaps we do need to have a consultant to help us be on the same page, and, you know, so that we can think more broadly or be on the same page of how we're thinking about racism.
Great. Andy, I see that you have your hand up.
Yes, thanks, Rick. I just want to follow up on the point Bob made, and also Gary. So, you know, as Bob pointed out, you know, all of us here on this call have been relatively successful in this system. One of the things we talked about in our working group calls is that, you know, their value in trying to reach out to those who have not been successful within the system, they have not received, you know, the grants, they have not received a job at EHS and so on, and to see their attitude and their perspective on the system might be helpful.
Andy, that's a really good point. And so maybe I'll throw on, kind of overlay this with the whole role of mentoring. So there may be opportunities for individuals who have been successful to be mentoring others who haven't been quite as successful of getting funding and working in the system. So I know that there's been quite a few discussions around, I think Gary mentioned this in his talk, about reaching out to certain underserved areas and seeing if we can do some mentoring to help to move them along. Edith, looks like you have your hand up.
Yeah, no, I was just going to push back a little bit, Rick, and sort of say, or, is it mentoring, or is it digging deeper and seeing if there's something about the system that needs to change beyond the mentoring? That, we have the system, so the people who are successful, we could either say that, okay, I'll mentor, or we could say, let's look a little bit deeper at the system and see if there are things that we need to change about that. So that's all I was going to say.
I think it's a good point, but why not do both?
Yeah, I'm not either or, but.
Yeah, and I think I would just, because it's been a frustration for me, is to try to understand, for instance, the funding issue, which is, that's the coin of our realm, is giving out money. Why certain groups do better than others. It's, and that was the motivator, I think, behind our efforts that Gary mentioned in his talk about trying to reach out to HBCUs who have historically not done very well in terms of getting NIH grants. So, yeah, I look forward to tackling that difficult issue. Yeah, Bob?
All right. This is actually Liam's suggestion, but Lou School on the P30 meetings we had, our anti-racism workshop event about a month ago, she actually came up with a list of consultants, so that might be a good place to start. And Liam has a copy of that.
Yeah, I think Gwen, you're aware of that, right? Yeah. Okay. Good. Always bringing consultants in the federal government with federal contracts is always a bit complicated, but I know Gwen has been working pretty hard on that. And I think Gary's been doing the same thing in DER too. So Mary, actually, did you want to say something, or any comments you'd like to add as part of a working group member? I want to make sure everyone has a chance to speak up. Go ahead.
Oh, no, I'm good.
Okay. Sounds good.
You.
So Karen, any other members of your working group who haven't had a chance to unmute? And
we haven't heard from Nicole.
Where's Nicole?
I'm here. Thank you for recognizing my efforts, and I'm just proud to be able to serve this working group. I have a lot of faith that a lot will come from this because the members of the group are clearly very passionate and have a lot of good ideas. So I'm very excited to be part of it.
Great.
I don't know that we've heard from Fred. You put something in the chat. Fred, did you want to weigh in?
We didn't hear from Fred.
Yeah, where's Fred? Fred
Fred is here.
Fred's usually not bashful to speak up. Go ahead, Fred.
I'm not bashful. One of the things, certainly this committee has not been able to impact yet. We haven't tried, but something I've seen across NIH, and even today, right now, the Common Fund first
I'm actually coming right now. I'm just locking up.
Is being reviewed. One of the things that's happening is I'm seeing that study sections are being populated differently than I've ever seen them before. When I say that, I'm seeing more African American scientists serving on study section panels. That I don't know if this is going to impact the outcome of reviews of applications that are coming in, but I think this is certainly a positive step that I have not witnessed before. And I guess I've been an extramural for about 22, 23 years now. And I've never seen, I've sent things to other people in review saying, geez, I've never seen review sections that look like this before. Today, there were so many folks on the panel, I couldn't even count them. So I think that there are some positive things that are beginning to happen across NIH, and again, I'm excited to be a part of this committee and see what we can do over the next couple of years.
Great.
I think we've heard from everyone on the committee now, so that's good.
So Karen, a question for you is, I went through the Unite committees and yesterday in my presentation on the charges. How does your committee, or maybe you haven't had a chance to talk about this, but how does your committee feel about the Unite committees? Does it pretty much cover the various things that need to be discussed?
We haven't talked about that yet. So we're still assembling the group, right? So we haven't talked about that yet. But one thing, of course, we don't want to reinvent the wheel, right? We want to work with others. And we've had that comment from several people now, and it's absolutely critical. However, I think when we our first jump into this, I think we would rather not be influenced by other people's organized ideas, would rather have our own and then see how they fit in.
Sure.
That was sort of the plan. So we haven't really discussed that yet. We all just sort of wanted to think, all of us agree we need data. We need data before we can begin, and we need data moving forward to evaluate our progress. That's one key. The other thing that I think we all agreed on was kind of breaking into the subgroups that seem the most relevant to us. And that's why I wanted input from everyone today. And I really like the input from everyone, from Katrina and Trevor. Of course, Edith gave, and Gary, a number of people gave feedback, but many of them are on the committee. But we heard from some that are not on the working group, and that's very valuable, and I appreciate it, to help us sort of to guide us. But I didn't want to really be influenced originally by other people's initiatives or organizational output until we begin. Then we see, of course, we're going back to Unite. That's what you want us to focus on. I remember our very first meeting when you asked me to do this, and you went through the Unite with me and said that's what you are interested in. And we will absolutely focus on those after we have an unbiased group organization attempt. And then we will see how that fits in, if that's fine with you.
Oh, actually, you're the chair of the working group. You're the one providing the leadership and the guidance on this. So whatever works for you works for me, and that makes perfect sense.
I'll point out that Gwen put a link to a blog from Dr. Burns from CSR related to equity and diversity in peer reviews, which I think is a topic we're going to have to wrestle with. So, I'm sorry, that the working group is going to have to wrestle with at some point.
Well, I put it in here because it was related to what Fred said. I think some of the things that they've been doing over at CSR are showing some results. And so you can read a little bit more about her vision and philosophy related to diversity and review.
Okay. So I really found this discussion to be quite stimulating. And thank you, Karen, and all the members of the working group. I'm going to turn it over to Rick now because we're at time. So Rick.
Well, being respectful to everyone, I want to keep us on time, and I want to adjourn right on time so we can go about probably to our next Zoom session. So, but let me close by just thanking Karen, all members of your working group, and then extending to all members of council. As I said at the outset, I understand it's an enormous time commitment to serve on council and on these working groups. I totally appreciate. I know that I'm speaking for all members of senior leadership and all of us here at NAEHS. We really appreciate your input, your engagement. And I also want to thank Gary. I want to thank Pat. I want to thank the staff in DERT. These meetings don't just come together in the snap of the fingers. There's been a lot of very careful preparation. I also want to thank Mary Jacobson and Spencer Smith in my office, along with Christine Flowers and Sheila Newton and others that helped pull together topics and slides for me. It's a lot of work, and so I totally appreciate all of the effort that's going on to make this happen. So I'd simply say that I think this is another example of, I'll say this is the best yet as far as meetings of council are concerned. So we plan to see you all virtually. I think it's going to be virtually in September, but
right, it is.
It will be in person.
Yeah, that decision has been made that September will be a virtual meeting. So.
Okay.
I also, Rick, want to just point, you said thanking people. I want to make sure that everyone realizes how much work Liz McNair and Rose Moody have done to pull this together. You all know who Liz is for sure.
Absolutely.
And I also have to give kudos to Nathan Mitchner and John Maruka, who are the ITAV people who are making this run. John's kind of stayed behind off the screen today. but they both do an incredible job. This is, I don't know how many times they've done this now, but and it's almost always gone without a hitch. So I just want to make sure they're recognized.
And I'm really glad that you brought that up. So there are so many people to thank, and you know, Nathan and John and others. Again,
I almost forgot our science writer, Ernie Hood, who does the minutes every round and does a great job too. So.
Okay, well, thanks everyone. And so in the interest of keeping us on time, we will see you all in just a few months. Have a great summer, everyone.